students. Hope all of you are fine. So dear children, we are doing the lesson cell cycle and cell division, and in this video we are going to discuss about the cytokinesis. Okay, type of cytokinesis in plant and animal. Okay, then cell plate method in the plant. Then we'll discuss significance of the mitosis. So let's discuss cytokinesis. What is cytokinesis? Cytokinesis basically deals with kaitos. Okay, in Greek means hollow or cell, and kinesis means movement. Is the division of protoplast of the cell into two daughter cells after the nuclear division or the karyokinesis, so that each daughter cell come to have its own nucleus. Cell organelle, mitochondria, plastid, Golgi bodies, lysosomes, endoplasmic reticulum, ribosomes are also distributed between the two daughter cells. Mitochondria and plastid undergo division by cleavage or by the fission. Details of the replication of other organelles are not known. At time of cytokinesis does not follow karyokinesis. It produces multinucleate condition known as synocytes or syncytium. Normally, cytokinesis starts towards the middle anaphase and is completed simultaneously with the telophase. Cytokinesis is different in animal and plant cell. Now we'll discuss first the cytokinesis in the animal. So in case of animal cytokinesis, the central equatorial part of the spindle gets changed into dense fibrous and vesicular structure called midbody. Simultaneously, microfilament collect in the middle region of the cell below the cell membrane. They induce the cell membrane to invaginate. The furrow deepens centripetally and cleavage the cell into two daughters, each having a daughter nucleus. The method is known as cleavage cytokinesis. Now, plant cytokinesis. The plant cytokinesis is different from the animal cytokinesis due to presence of a solid cell wall on outer side. It takes place by two methods, cleavage and cell plate. So number one, cleavage method. It takes place usually in the some lower plant. Cytoplasm undergoes centripetal constriction in the middle to form two daughter protoplasts, each having single nucleus. In the furrow between two protoplasts, pectin, hemicellulose and microfibrils of cellulose are deposited to form double wall. The wall development is centripetal like the cytoplasmic cleavage. Now next we have is the cell plate method. It is common method of cytokinesis in the plant cell. In each case the spindle persists for some time. It is known as the phragmoplast. Okay, so phragmoplast is formed in case of the plant cell division. Small vesicle produced by the Golgi apparatus collect at the equator of the phragmoplast. The membrane of the vesicle fuse to form two sheets which enclose matrix or the film. Soon the film becomes solidified to form the cell plate or the middle lamella. It grows centrifugally and come in contact with the lateral wall of the parent cell. With the formation of cell plate, the spindle or the phragmoplast disappears. The daughter protoplast deposits cellulose, hemicellulose and pectin on either side of the cell plate. They form the primary wall. Now we'll discuss the significance of the mitosis. Mitosis help in growth. Somatic cells are formed by mitosis. Therefore, mitosis is essential for growth and development of multicellular organism. Human baby has about 6 into 10 to the power 12 cells. All of them develop from single cell zygote. Through repeated mitosis, plants are able to grow throughout their life due to mitotic cell division in their apical and lateral meristems. Maintenance of the surface and volume ratio is another function. On an average, overgrown somatic cell is induced to divide so that mitosis help in maintaining a proper surface or volume ratio. Next, an efficient cell has a high nucleocytoplasmic ratio. Increase in the size lowers the ratio. It is brought back to the efficient level through the division. Next is mitosis involves replication and equitable distribution of all the chromosomes so that all the cell of the multicellular organism have the same number and type of chromosome. This help in proper coordination among different cells. Regeneration is another property. Mitosis keeps all the somatic cell of an organism genetically similar, resembling the fertilized egg. They therefore are able to regenerate part or whole of the organisms. Now, mitosis is the method of multiplication of the unicellular organism, that is the reproduction. It is mechanism for replacing old or worn out cell. In human body, roughly 5 into 10 to the power 9 cells are daily lost from the surface of the skin lining of the alimentary canal, RBCs, WBCs, etc. The same are replaced by new cells formed through the mitosis. Healing: an injury or wound is healed by repeated mitotic division of the surrounding healthy cells. Now, mitosis provide multicellular condition. It provides opportunity for differentiation. Uncontrolled mitotic division lead to cancer. Okay, now evidence of basic relationship. The detail of mitosis are similar in the majority of the organisms, showing their basic similarity and relationship. Dear children, you can see some differences between the plant cytokinesis and animal cytokinesis. You can see some differences between plant mitosis and animal mitosis. Some more differences between the plant mitosis and animal mitosis.